Hello, today I'll be teaching you endospore staining. So let's start. How to write it in practical record? You need to write the aim, introduction, principle, requirement, procedure, observation, result. Okay, let's start. To demonstrate the spore structure in bacteria and the presence of endospore. So endospores are present in some bacteria, and endospores are typically inactive, dormant, and they don't reproduce. So the few key points are important about the endospore, which is like endospores are resistant to heat, radiation, chemical and other agents. So it is typically lethal to organism. Why it is highly resistant to heat? Because of high concentration of calcium ions and dipicolinic acid. So what happens? These vegetative cells form and produce the endospore during the sporulation. Let's see. See, this is the vegetative cell, and it reproduces during the binary fission. But we'll be seeing here the sporulation procedure. So under unfavorable condition, it starts the sporogenesis and endospore. So you can see this color, green color is the endospore, and this red color represents the vegetative cell. This is just coloring to show you what is the where is the vegetative and parent cell and the endospore. Okay, so endospore is formed. After that, the vegetative cells start degenerating, and only endospore will be left with the spore coat. And again, after a long time, like if it will be there under favorable condition, so again it will germinate and become vegetative cell. So the spore coat will rupture and it will transform into again vegetative cell. So the main thing of showing this sporulation is about the endospore formation. See, when you do the endospore staining, you will be able to see the endospore location also in parent cell. So what are these locations? See, these are the central one, the centrally located. You can see the endospore is located at the center, and these are the subterminal one, and these are located not at the center, not at the end, but between that. So that's why it is called the subterminal location of the endospore. And these are the terminal. You can easily see these are present at the end of the parent cell. Okay, so this is really important to remember these locations. And now we will start the procedure. I'll be showing you with the diagram. You need to take the clean glass slide, and you have to take the 48 hours or 36 hours old culture. Then there is a distilled water, a drop of distilled water. Take an inoculating loop, take the pre-grown culture and make the smear and heat fix the smear. Okay, after heat fixing, what you need to do, you need to take the malachite green stain and safranine, and also you need to start the water bath. See, this is the water bath, you can able to see, and you need to keep this slide on the staining rack which you have kept under the water bath, and then you need to flood the slide with malachite green for 8 to 10 minutes continuous. Do not let it dry for any second. Continuous flood the slide. So what happens when you add the malachite green? It penetrates under the spore coat. Okay, then wash the slide with distilled water drop by drop. Then after drying, add the safranine stain, okay, for 30 seconds. Then again wash it, the excess stain, with distilled water drop by drop, and let it dry or dry it with blotting paper, and then observe under microscope. So why we have used the safranin stain over here? Because vegetative cells, the parent cells will be there, that will be stained with the safranin, not with the malachite green, because malachite green has washed away after the distilled water washing. So that is why we use the safranin stain. So this experiment shows a really beautiful observation. I personally really like it to see under microscope. So you can see my observation. This kind of structure you'll be able to see under microscope. Exactly this kind of structure. You can see the rod shape of bacteria and this green color, because this pore coat has taken up the malachite green stain, and this is the typically endospore and it is at the end. So this pink color cells you can be able to see because of the safranin stain, and this is the located terminally and this is centrally located. So this kind of observation you can easily be see under like 100x oil immersion. Then you need to write down the observation table. So again the given culture and then observation, the shape and the location of the spore and the result. So green colored endospore appeared. So this is all about the endospore staining. Next topic will be on capsule staining, and if you like my video, please do like, subscribe my channel and please share with your friends. Thanks for watching. Bye.